Peace. Hi, everybody. I think we should start. The communication team, please start with music. Thank you. Can you hear me all right?
Yes, I can.
Great, Musa. Thank you. Hello, everybody. Good afternoon. Happy Sabbath. I just want to welcome you to this short discussion, which I trust and fully believe that will be profitable to you. By no means will we, cover everything today, but at least this can give you some information which might be useful, and we can have other following conversation later to just build on the subject. So what I'll do, I'll just read a verse, then I'll pray, then I'll invite our guests to lead us. So I'm going to read from the book of Hosea, chapter 4, verse 6. Maybe it's a little out of context, but it talks about knowledge and its importance. So Hosea 4:6 says, "My people are destroyed for lack of knowledge. Because thou hast rejected knowledge, I will also reject thee, that thou shalt be forgotten." Sorry. From the book of Hosea, chapter 4, verse 6. Yeah, can we mute if we are not talking? Can we please mute? Okay, great. Thank you. Yeah. "I will also reject thee, that thou shalt be no priest to me, seeing thou hast forgotten the law of thy God. I will also forget thy children." So here I'm just trying to apply this verse in a very broad context. Nature also has its laws, and God has given us a lot of information by which we can live by and be safe. So I want to pray now. Then I quickly allow our guests to come in. So let us pray. Father, our Lord, our God, who art in heaven, we thank you for being with us until now. As we go into this program to discuss about COVID and the existing knowledge so far and what is not known, we pray, Father, that you might be with us and talk to us and minister to us. We pray this, trusting and believing, for we pray through the name of Jesus Christ, our Lord and Savior. Amen. Yeah, so I want to introduce our guest today. His name is Musa Mulongo. Musa Mulongo is a good friend of mine. So when I approached him, I only gave him an option to say yes. Musa holds a PhD in immunology. He also serves as a senior scientist at ILRI. So he's very well versed with the technical aspects of this disease. and I hope we will profit from his knowledge today. So what we'll do, the process will be Musa will give a presentation. Then after that, we will take questions and then discuss on those. I hope you have noted down all your questions, and during the program, during the this discussion, you are also free to list other questions which you want handled. I'll also, in my small capacity, offer Musa some support from a technical side of things. Thank you. Musa, do you want to say hi and you take over?
Yes, but you need to give me the right to share my slides.
Oh, this needs to be our communication people, yeah?
Yeah, I cannot share the screen unless I send it to you.
No, even I don't have the rights.
Ah,
so, yes,
okay.
yes. I'm just a guest. So maybe you can switch on your video, usalimie wasafiri wa mbinguni.
That is fine as long as, so do we have a way of sharing the slides?
Yes, yes.
Okay, good.
We'll try to.
Okay. Thank you. So, yeah, Tom has indicated my names, and I'm also a Christian Adventist, like all of you.
Amen.
And I am delighted to join you this afternoon to talk about these things. On the top of my slide, which I will be sharing once this is sorted out, I have indicated that I've taken out a quote from the Book of Patriarchs and Prophets, page 115 and 116, where Sister White says that those who take the written word as their counselor will find in science an aid to understand God. And so, as Christian Adventists, we do not just do science and like others, but we take science as an aid to understanding God, meaning that God has given us knowledge, and whatever we do with that knowledge in science ought to be to the glorification of his name and to share with humanity. And so through science, we also understand God. It is important to ground that truth in the presentation that we are going to have today. So Tom, if you let me know when I can share, I will go ahead the screen.
Yeah, I'm still waiting.
Okay, I see that I can.
Okay, good.
Whoever has done this in record time, thank you very much.
Sante.
So, yeah, good. Is that clear to everyone? Please, if you're not seeing the screen, let me know.
It's perfect. Okay,
so I'll just go ahead. It's the problem with these online meetings is they lack the warmth of fellowship that we would have together, but that's fine. We'll still go ahead and learn. So yeah, so I started by indicating that those who take the written word as their counselor will find in science an aid to understand God. And that is the context in which we're having this teaching. So I'm going to tell you what I'm going to tell you, and then I will tell you, and then I will tell you what I have told you. And so if you forget everything, I think these are the key messages from today's message. And I've tried to make the presentation as simple as possible. I've gotten most of my illustrations from popular, normal media without going into crazy scientific detail. So one, infectious diseases that cause pestilences and pandemics are as old as recorded history, and most of them are a result of animal sources. Secondly, we do not know the exact origin of COVID-19. Scientific experts believe that it was not made by man in the lab. 3, vaccines are the most important discovery of modern medicine, and they are mostly safe and are licensed after comprehensive safety and safety assessment. And then finally, we should do our best to protect ourselves and our loved ones, since the pandemic is going to be here with us for a while. it's important to underscore the last point that the nature of pandemics is they get into the population and they get going. So it's going to be here with us for a while. It's noteworthy to say myself, I'm a survivor of this pandemic. I stayed in hospital for 17 days and came out last month. And I can attest to the fact that it is a severe infection. So just to get going then, this is the history of pandemics. The slide that I'm showing you, since recorded history, we have had quite a number of pandemics, with the biggest being the Black Death of the 1300s. It lasted about 4 years and it killed about 200 million Europeans. It was caused by a bacterium called Yersinia pestis. It came from mice. And you can look through the time, the ages, so to speak. You've had smallpox, you've had several other plagues, you've had a cholera outbreak that has killed a number of people. The HIV/AIDS pandemic is still with us. And then, of course, you had the major, most recent one, the Spanish flu, that killed up to 50 million people just after the First World War. And then, of course, the other small ones, swine flu, Hong Kong flu, and others. So when you look at COVID-19 in the context of history, it is almost nothing. This was recorded around the end of 2020, around March 2020. It had only killed about 7,000 people. Now we are looking at 2-3 million people, but nothing close to the scale of the older pandemics. This is just to tell you that these bugs live with us. They are with us. We can say they're a result of a sinful universe, because God created a perfect world, but then these things still happen. I'm showing this slide specifically to make the argument that there's lots of conspiracies around this was made and given to us and things like that. But the truth is that as years, as old as you can go back from the Justinian plague and all that, nobody knew even that viruses or bacteria caused diseases. So they couldn't make them, but we still had pandemics. That's important to underscore. Just as an example, again, I've mentioned the top two, the Black Death of the 1300s and the Spanish flu. Those are pictures. So the Black Death, if you look at the illustrations, people are dying everywhere. And because they did not know that diseases are caused by pathogens, they thought that angels of death had come to harvest people at that time. And these are the drawings that you see from that time. And then if you look on your right, the Spanish flu, more recent, people are putting on masks. You have all those isolation tents out there and people getting treated, meaning that the point I'm just making is that pandemics are with us, some with varying severity, and we shouldn't feel any special as people living in this time and age that this is happening to us. And so where did this bug come from, really? The first human coronavirus was first identified in 1965. It seemed to just cause a common cold, but then as time has gone by, quite a number of different families of coronaviruses have been discovered, with 7 types that affect humans and many other animals like the bats, the pangolins, raccoons, and others. And people think that the current COVID-19 virus originated in bats, just as you have 2 other relatives of this one, the severe acute respiratory virus that emerged in southern China about 20 years ago. It did not kill very many people, 700, infected about 8,000, just causes a fever, headache, respiratory problems, and then just almost like COVID-19, but COVID-19 is more severe. And that's why we call this SARS-CoV-2, because we also have the earlier SARS coronavirus 1, which is the SARS one that happened in 2002. And of course, you also have the Middle East respiratory syndrome, the virus that caused that. It was also another coronavirus in Saudi Arabia, killed just about 800 people, did not infect too many people. Same as SARS in terms of the disease it causes, but also caused kidney failure. And like I've said, scientists have not found any evidence that the virus was made by man. There are certain specific clear signatures that you can be able to find if you are trying to discover or find out whether a bug has been developed in the lab or not. In this case, there is no evidence of such signatures, molecular signatures in this virus. So when you think about it, it is not strange to think about a situation like the picture that I'm showing you, where the city from which the virus is thought to have begun in China And they have all these kinds of mix of wild animals and plants together in a wet market. Wet market just means they bring the animals, wash them there, kill them, they are fresh, and then sell them. And so you have lots of different animals, including snakes and all that, in this crowded place. Most of these have been captured in the wild, and people come there and buy them for the purpose of meat. And so you can imagine if you go to a normal abattoir in Nairobi, for example, where we kill animals, a butchery, let me call it that, but where we kill the animals before taking the meat to the butchery, you'll find that the place smells a bit weird, and that's just cattle meat. You can imagine now if you bring all these kinds of species into this market and wash them and kill them for food. It's crazy. You just have a mixing and melting pot of animals and pathogens spreading around, and it is suspected that this is how this pandemic started. So that's just a quick introduction to the pandemic and how does COVID-19 spread. I just put up this slide because people say, well, I was someone who's had COVID-19, I didn't get sick. Others say, I am caring for a patient with COVID-19. I didn't even know they were sick. I didn't get sick. Why is it targeting some people and not others? But when you think about it, really, there are many factors that cause you to get an infection or not. It is the type of virus. You've heard about variants, and we'll talk about them later on. Is it less infectious or more infectious? Is the status of your immunity okay? Are you immunocompromised or are you healthy? Do you have enough sleep? Do you have enough exercise? How is your immunity? How old are you? The stage of the disease. If you have a patient with more symptoms, coughing, crazy fever, of course, they are likely to be shedding more virus. Shedding just means they're breathing out more virus through their mouth or through their nose. So you have a high viral load that is being released into the environment. If you breathe it in, it's likely that you would get sick. And then, of course, distance from others. And if you look at this picture, the ways COVID-19, that is referred as SARS-CoV-2 here, spreads is much or more or less the same as influenza. The R value is really saying if you have one person in purple, if you look at this cartoon that I'm showing, they're likely to infect 2.5 to 3 people really statistically, so that not all the people around them will get infected. And we use the R value to estimate just how quickly and how infectious a pathogen is. So for COVID-19, I think it's about 3 or 4. So one person can infect about 3 or 4 people that they come into contact with closely. And all these factors determine whether the person nearest to this person might get sick or not. And so we just use what we call an R value of 2. For example, I'm giving an example here that if you are one person looking at the cartoon on the right, you will infect 2 other people, who will infect 2 other people, who will infect 2 other people, or 3 other people, and that is how the infection spirals out of control. So I know that the most interesting discussion in the room really are vaccines, and I'm going to spend a little bit of time to talk about what vaccines are, and I will just use COVID-19 as an example for everything that I say so that we can have a robust discussion afterwards. So I'll just describe the concept of vaccination in really simple terms. You have a pathogen that causes disease. It is made up of many different proteins and structures. And when you get an infection and this virus gets into you, your immune system sees different parts of this virus. Different cells of your immune system see different small molecules on this pathogen. And what we do essentially when we are developing a vaccine is we take one little part of that virus that we know your immune cells recognize, and we produce it separately from the virus, and we inject it into you, and you develop antibodies to it, which bind those particular particles. Nonetheless, there are different ways of making a vaccine. I'm only describing the COVID-19 vaccines, and I'll go into detail, but you can still kill the whole pathogen and inject it into you. It is dead, it will not cause a disease. You could kill it with the heat or other chemicals, and we can inject it into you, and you'll still make some responses, and you will get protected, and I will go through what I mean by protection. But essentially, the point here is that we do not inject you with the virus and cause disease in you when we are vaccinating. All we do is we take a little part of the virus that does not cause any harm to you, and we put it into you, and you have antibodies that are produced to it, which are supposed to protect you, and I'll describe what that exactly looks like. So in the case of COVID-19 specifically, did someone speak or I go on? Okay, thank you. So in the case of COVID-19 specifically, we have these spike proteins that sit on the surface of the virus, and it's called a coronavirus group of families because they have crown-like structures. And that's why they're called coronavirus, just because it's a crown. And it is these spike proteins that give the virus that shape. And what happens is that you have the spike protein that sticks out, and this spike protein binds to your cells. It could be lung epithelial cells. I don't want to complicate it, but just binds to your human cells. And the goal of vaccination is, like I have shown you previously, is to be able to isolate this spike protein on its own and inject it into you so that we generate these antibodies, and the antibodies will be generated, bind to that protein, and that will be it, so that when you have an infection next time of the whole virus, which is expressing exactly the same kind of protein, your antibodies will bind to these spikes and stop this virus from binding to your cells on this receptor and getting into your cells and multiplying and causing disease. So essentially, the whole point of COVID-19 vaccines is to create new antibodies to the spike protein of the SARS-CoV-2 virus and block the virus from entering human cells. And through this process, the virus will be killed because it has to bind to those receptors in human cells, go into the human cell, and multiply itself because it has food there, the environment is good. But if you to bind these proteins, these spikes on the surface, then the virus basically just dies. So basically, this is the whole concept of not just COVID-19 vaccines or all vaccines, why we vaccinate. We first of all have what I'm calling a primary immune response, where we take that protein that I've described, we inject it into you. You do have an antibody response that lasts for a short period of time, and then the antibodies go down when that molecule is cleared from the immune system. And then when the real infection comes, that we are calling a second exposure, you have this explosion of an immune response, so that you have antibodies that are released in large quantities, and what they do is their goal is to overwhelm the immune response, and then you are able to control the infection. That is the whole basis of all vaccination. And importantly, your immune system is able to develop memory. So the whole reason why we do this is just to trick the immune response to remember that I have dealt with proteins that look like this. Now I have other proteins that look exactly like this, and I'm going to respond faster and be able to clear out the infection.
Okay.
Hello?
I think someone is on the phone. Can we mute if we're not talking?
Thank you. I think that's done. Yeah. So that's the whole point of. Okay.
Okay, I see the phone is now muted. Thank you.
Okay. So just one little lesson in vaccines, because it will really help us to understand the next stage of this discussion, is that we need to understand when people talk about DNA and RNA and protein and what that means. I'm going to make this analogy. If you remember your lessons, DNA is the genetic code or the language that keeps all instructions for the formation of and the function of all life, whether it is plants or bacteria or humans, or it's just 4 molecules that are combined in different ways. And it is where the instructions for life, essentially, and life's processes sit. And that is the DNA molecule. However, the DNA molecule if you look at your blood and you look at your skin and you look at your nose, you're not looking at DNA, you're looking at lots of proteins. And those proteins have been instructed to make your nose or your eyes or your hair or whatever it is about you in a specific way by what is in your DNA. However, we just don't have proteins instructing other parts of molecules in your body. We don't have DNA just making instructions so that protein is formed. You have a translator in the middle, which connects the language from DNA to the protein, and that is what we call the RNA. So if you think about a house, just to simplify this and break it into its most simplest form, the architect draws the house. And he says to the engineers, I'm giving you a drawing of the housing, build this house. And the engineers figure out and work on it, and you have a complete house on your right, if you look at the cartoon that I'm showing. So DNA is like the blueprint of the house, the guidelines, the directions of how to make that house. And the engineers bring that blueprint into life. And then you have a house that is made, that is built in. So DNA is like the drawing of the house. RNA is the process of making that house, and the protein is the complete house that has come out. And this will become clear and important shortly, because we'll have questions on RNA vaccines, and all these stories about RNA vaccines come and get into our DNA, and then basically they are going to change us. And that's important for me to point out. And finally, the immune system only responds to proteins. It could respond to RNA or DNA in mild ways, and we can discuss that. It's more complicated. But eventually, the most important thing is that immune response is always to a protein, and not to a DNA and not to an RNA, to a processed clear protein that is foreign. And then it is recognized as foreign through danger signals that it shows. And then you have cells of the immune system that start fighting it. So I'll just go straight to the vaccines and just have a quick description based on the background that I've given you on how basically RNA vaccines work, be they from Pfizer or Moderna for COVID, or even if not for COVID. Essentially, what scientists do is they take the DNA from the virus, the DNA that is responsible for making the spike protein that I have described, and they let that DNA become translated into RNA so that it is able to, the RNA has instructions to make viral proteins. And this is formulated into small particles that are protected by a lipid, don't worry about that. And then this is injected in you as a formulation, and you release antibodies. The RNA is released into the cell, and what happens is that your own cell starts to make proteins of COVID-19. But those proteins are not harmful to you. Those proteins will not cause disease to you because they're just a little piece of the whole function of virus. And so your antibodies are produced to eat the little things in green that you see on this cartoon. And then in that sense, if you have a real COVID-19 virus infecting you, these antibodies protect you. There is no time when this RNA becomes DNA again and sits in your DNA and starts changing you to become a monkey or a crocodile or anything else. Just the same way the engineers building the house do not go back and start redrawing the house so that they remake the house. It is a process. And so what we do with RNA vaccines is we start somewhere in the middle where we just translate as little sequence of RNA, of DNA into RNA, and then we give you that, and your own body produces antibodies to it because those molecules are recognized as foreign, the proteins that have been produced. And then that is wiped out from your body within no time. The RNA is not self, it doesn't sit there, it will be cleared out in a number of days. That's the whole basis of RNA vaccination. Going back to now the AstraZeneca Oxford vaccine, the basis for that is a little different. The concept is the same, to generate antibodies against the spike protein. However, the approach is a little different. What they have done is they have taken a chimpanzee adenovirus. Adenoviruses are all with us in nature. We all have them, human adenoviruses, chimpanzee ones. They don't cause disease largely. In us, So we carry them, and all they have done at Oxford is taken one of those viruses called the adenovirus. They have taken it, and then what they have done is they have looked at the SARS-CoV-2 virus again. They have taken the DNA of the spike protein. They have put it into the virus so that it is protected and carried there. And eventually what has happened is that virus is injected into you. It multiplies, it doesn't cause any disease, but it keeps on making copies of the spike protein of COVID-19. And once that is expressed, then obviously you start producing antibodies, and then you are protected if you have an infection with the real virus. That is all we do in vaccination. This has the two principles of the Oxford AstraZeneca vaccine and the Moderna or Pfizer vaccine, which are the ones that are currently being sold. So the point I'm making is nobody is infecting you with a virus. Nobody is giving you something that should harm you. The goal is for your immune response to be sensitized and know that I have seen this molecule before, and next time you have the whole virus, you have an explosion of a response. And so, like I've said, it's important to say RNA vaccines will not mix your DNA and change you, because RNA vaccines are like the structure that engineers are building, and you have a whole house as a protein out of it. It will not go back into a house that is a blueprint that an architect has drawn again. When you look at this diagram that I'm showing you, you have DNA being copied into RNA, which is translated through complex processes into a protein. For humans, this is only one directional: DNA, RNA, protein. In viruses and other plants and other molecules, you might have an interchange between RNA and DNA, where you can have RNA becoming DNA, and then that DNA becoming RNA. We call it reverse transcription process. We have no molecules or enzymes in humans that can translate RNA to DNA. That is why when we give you a molecule like this, we are completely confident that this is not going to sit into your genes, into your DNA, and start causing other issues. And there's lots of work around RNA vaccines before these first ones that have been produced for COVID-19 that demonstrate that in monkeys, in mice, and other animal models, we do not have RNA being converted back into DNA, at least for higher animals like us and primates. So these are just a few of the attributes of the vaccines that we have right now in the market. I think there are 4. You have the Pfizer vaccine and the Moderna vaccine that are both RNA, and they provide 90% and 95% protection. I'll describe shortly what that exactly means. And one is stored at -70 degrees and the other one at -20 degrees up to 6 months. And this storage is just about how they have been prepared and formulated and tested and stabilized, because RNA is not a stable molecule. It always has to be stabilized and protected because it degrades very quickly. And then you have the Oxford and the AstraZeneca, the Oxford AstraZeneca vaccine and what we call the Sputnik vaccine from Russia that you've been hearing about. They are both viral vectored vaccines, exactly as I've described. DNA taken from that little spike protein of COVID-19, put into a virus that does not cause disease in you, injected into you, and the virus multiplies, and you have copies of that protein produced, and then you have an immune response. And those two are produced at regular fridge temperature, 4 degrees. This is really important because countries like ours, which don't have systems, freeze things up to -70 or -20 constantly, going to the field, then can be able to carry them just in freezer boxes. So I need really to spend a few minutes to describe this slide on what the main goal of vaccination is, because there's really mixed messages around this and people get confused. The reason why we vaccinate is not necessarily to stop you from coming into contact with the virus. You'll come into contact with the virus. But the reason why we vaccinate and the benefits of vaccination is, one, we try to reduce the severe development with the development, not the severe, the development of severe clinical signs of COVID-19 symptoms, basically the crazy COVID, coughing, the uncontrolled fever that comes with it, and the collapse of your lungs so that you are not able to breathe. So when we give you the COVID-19 vaccine, the goal is to reduce severe infection or severe disease from COVID-19, clinical signs. The second goal is to reduce the length of time from catching the disease to recovery. So you are vaccinated, you come into contact with COVID-19, we are trying to make you cough for 2 days instead of 10 days, basically, so that you have a reduced, you feel that, oh, this was a common flu. That's our aim. Thirdly, is we want to reduce the likelihood for you getting hospitalized, because if you're having less severe symptoms, then we all don't go to hospital when you have common flu. And that is the goal, that you just feel you have common flu, you don't get hospitalized. We also vaccinate you to reduce the likelihood of death, because if you have, of course, less clinical disease, it's unlikely that you are going to die. And we also vaccinate you so that we reduce the probability of you infecting other people. Importantly, vaccination does not stop the virus from entering your nose and mouth and staying there for a while. This is very important, and I think that the way messages around this have been packaged are not helping the public. You can get vaccinated, and when we do the swab at the back of your throat or nose, we might get COVID-19 virus particles, which we will use that PCR diagnostic method to amplify, and we'll say, oh, you have COVID-19. Having COVID-19 sitting at the back of your nose and your mouth does not mean that you are sick with COVID-19, unless it is causing a cough in you, unless it's causing breathing problems in you because it's affecting your lungs, unless it's causing fever in you. Clinically, we are able to say that you have the virus, but you are not showing clinical signs. And we are saying that vaccination might not stop you from breathing in the virus and having it sit there for a while as your antibodies do their bit to clear it out. 2, vaccination does not stop you from infecting someone else, because if you have breathed it in and you're in close contact with someone, it's possible that, yes, you have protected yourself, not showing much disease, but someone else might get sick. And that's one thing that vaccination does not stop. And finally, vaccination does not stop you from getting sick from a new variant, but reduces the likelihood that you will get severe disease from the new variant. So the concept of variant is very simple. I'll just go to it straight away. When pathogens infect us, be they bacteria or viruses, a race begins between those pathogens and our immune system, so that we respond to very specific patterns on the pathogen, like I've shown you. But as the virus continues spreading amongst us, it starts to try to avoid our immune system by changing the patterns that our immune system is recognizing. And so then you have suddenly a slightly different virus, and then our antibodies can't bind as well, can't block that infection as well. And then people start saying, well, this is a new variant. We have to find a way to make a vaccine to it. We cannot make vaccines against variants that we have not seen. If you look at the concept, and we have no way of predicting what variants, what new patterns the pathogen will come up with to avoid your infection, to avoid your immunity. But the most important point is that when we vaccinate you, you might get sick from a new variant that is a little bit too far from the pattern that you are vaccinated for, but you will still retain some measure of immunity. And that's why you hear people saying the AstraZeneca vaccine protection reduces to 50% against new variants, which just means that out of 100 people, it seems that 50 people seem to still have some form of clinical disease. But we do not have any evidence now that someone who has been vaccinated or someone who has had the infection has gotten severe disease from a new variant. That's important to point out. So the goal of immunization, again, is this is what I'm describing here, is the concept of herd immunity, where these little people, the ones in blue are healthy people, but they are not immunized. The ones in yellow are immunized and healthy people. The ones in red are not immunized and they're sick and they're infecting other people. The goal of vaccination is to move from as many red people as possible to as many yellow people as possible who are immunized and who are healthy, and who, because of that, they are going to be able to protect others. So we want to have as many people that have been immunized and healthy as possible, so that they come into contact with, when they come into contact with people who have not been immunized and are healthy, those people will not get sick. And then that way, we will completely reduce the burden of infection in our communities. I'm going to end this talk by giving you a small overview of really how vaccines are developed. Vaccines are normally developed historically. Traditionally, it takes 7 to 10 years to produce a vaccine and take it to the market. And we start in the lab. We do what we call studies that we test hypotheses. We ask the question, what part of this molecule does people, that does B cells, or forget about, do immune cells of an individual see or respond to? And once we do that, we do some preliminary work. Then we look at whether the molecules that we are getting, the proteins that we are getting, cause toxicity or not. We study toxicity in mice. We look at where they go. Do they go to the spleen? Do they go to the brain? If they go there, what happens? And we discard very early the ones that seem to be problematic. And then we start going into what we call clinical trials, where we go to healthy people, vaccinate them, and then look at how they respond to the vaccine. And then we increasingly start increasing the number of people that we test until we get to what we call phase 3 clinical trials, where you have thousands of people tested. And then we continue looking at data that comes out of this, looking at, were people protected? Did they get sicker? What happened? And so if you look at this, COVID-19 vaccines under normal circumstances should have taken about 6-7 years, but this was a very accelerated way of developing vaccines because of the fact that it was a pandemic. It was stopping life as we know it, and tons of money was poured into this. As you can imagine, this is very expensive to do. So the reason why we have this accelerated vaccine development timeline from 6-7 years to 1 year in which we have developed a vaccine is just 3 good reasons. The first one is that we know the kind of virus that it is because we have dealt with it before. So we are not starting from scratch. 2, we have a lot of investment that went into it at the same time, with many companies trying to do this at the same time. And the third reason, I think, was the urgency around it, which I think is not a third reason. There are two main reasons. No, the third important reason, is really important, is advancement in technologies so that we can actually formulate RNA technologies this way. 10 years ago, 15 years ago, we would not be able to do this, but we have advanced so much that we can be able to do some of these things in a much shorter period. Those are the 3 reasons why we have this accelerated vaccines for COVID-19. And like you can see from this cartoon, data or information coming out of these trials has been reviewed on a rolling basis continuously to be able to get all the way to licensing. So typically, and I'm about to finish, the process of licensing a vaccine is very extensive. We start with a phase 1 trial, 20 to 100 volunteers. We ask questions like, is the vaccine safe? Does it seem to work? Are there any serious side effects? What size of the dose is required? We look at phase 2, where we increase the number of people to several hundreds. We ask, are there any short-term effects to this? Are there any long-term effects if we have the time to study this? How are the people's immune response responding to this? Then we go to thousands of volunteers in phase 3. We ask questions whether the vaccine is effective at population level, and we are able to move that into a licensing process. And there are experts and people who sit and make sure that the trials have been done properly, the molecules have been formulated properly. If you look at any vaccine right now for COVID-19 being sold, you can download the package and it can tell you exactly and in detail what is in those vaccines. Finally, and this is a big question in the room, are vaccines safe? Are they 100% safe or not? The answer is that the vaccines are safe for the majority of people. When we say the vaccine will protect 95%, we are saying that out of 100 people that were vaccinated, 95 seemed to be protected from catching, if it is COVID-19, severe disease. That is important. And 5% of people seemed to catch the disease. That's all we mean when we say 95% protection or efficacy. And so for the 5% of people who will still catch COVID after being vaccinated, the truth is we have no idea why they still catch COVID after being vaccinated. We are limited in our capacity to predict who will be safe and who will not be safe. And therefore, vaccine licensing is based on the number of people who have been protected by the vaccine in trials, which means it's a high number, then the vaccine is licensed. If most people are showing mild symptoms, side effects, then we allow the vaccine. If many people are showing severe side effects, we don't allow the vaccine. And we also try to look at the people who have gotten the vaccine in the trials, did they have pre-existing conditions or not? Overall, vaccines decisions are made on the basis of what is beneficial to the majority of people. And we'll get to that discussion shortly when you people ask questions about the AstraZeneca vaccine and blood clotting and things like that. So I think that you have, we're just about to finish. These are some of the questions that people ask. I will not answer them. I will listen to you as you ask some of them. But people ask, what are new COVID-19 variants and can vaccines protect us from these new variants? I think I answered that to say that, yes, you'll have a measure of some partial protection against these variants. It's highly unlikely that you'd get severely ill and die after you've been vaccinated if you get a new variant that is circulating. Why have COVID-19 vaccines been developed very quickly compared to vaccines for other diseases? I think I've answered that. What is the truth about blood clots in people vaccinated with the AstraZeneca vaccine? I think this is a question that has come up in the last 2-3 weeks. And I got some statistics that out of 5 million Europeans who are vaccinated with the AstraZeneca vaccine, 30 people, women under the age of 55, showed or experienced these blood clots, which is a percentage of 0.00022 of the people who are vaccinated. So if you look at 5 million people and 30 people showed those reactions, it just means that the vaccine is largely safe to the majority of people. And most importantly, people are struggling in the scientific world to find out whether even these 30 people who showed these clots are a result of the vaccine or not. So far, there is no proof that that came from the vaccine. In the UK, 11 million people received the AstraZeneca vaccine, and out of this, 11 women developed these blood clots. And that is a rate of 0.0001% of the people who are vaccinated. And like I've said, we have no proof that that particular vaccine was responsible. And the last question is, people ask, can I still get sick after being vaccinated against COVID? I think we have dealt with that. You could get very mildly sick, but eventually it's highly unlikely that you will be hospitalized. And so we can reasonably say that after vaccination, you will get, if at all, very mild clinical disease. I want to stop there and allow you to ask questions. I have told you that infectious diseases that cause pandemics and pestilences are as old as recorded history. Most of them come from animal sources. We do not know the exact source of origin of COVID-19. Scientists believe that it was not made by man in the lab. I've told you that vaccines are the most important discovery of modern medicine, and I could have shown you many graphs that show that disease incidence or the frequency in which we have diseases in a population, even for childhood diseases, drops considerably as we vaccinate more and more. And I can show you, and you can get from literature graphs from 1970, 1960s to show that we have saved many lives, especially for young children, through vaccination. And I've told you that vaccines are mostly safe. They are licensed after comprehensive safety assessments, and we should do our best to protect ourselves and our loved ones since the pandemic is going to be here for a while. Thank you very much. I think we'll take a few questions. and you are welcome to interact with me. Those are my contacts. I'll be happy if you use the Gmail address, and then we can talk about it some more. Thank you, Tom. I hand it back to you.
Thank you, Musa. This is, I think I really enjoyed your presentation. I hope it didn't fly over the heads of of the listeners. You know, I could follow everything because I'm a scientist, but I hope it was simple enough for everyone else, and that's why I think there is merit to open this session for questions, so that everyone can clearly understand and also be able to share this knowledge with other people. So one point, I'll just make one point, then I'll allow Olivia. I see you've raised your hand, Olivia. So as Musa said, I think it's important to distinguish between COVID infection and COVID disease. Yeah, these two are not the same thing. Yeah, so even after vaccination, you can still pick up the infection but not develop COVID disease. And I think I was reading this week how these vaccines performed in terms of COVID disease in phase 1 to 3 trials, and they showed that none of the individuals who received the vaccine in trials developed COVID disease which needed hospitalization. So I think that is very encouraging statistics. So I will allow Olivia to raise your question. Then next will be Elda Mube. Thank you. Olivia?
Thank you for the opportunity. Thank you, Tom. Thank you, Musa, for the nice presentation. We have learned quite a lot. There's been talk about, I know you talked something about immunity, about herd immunity, people saying that once you've been infected with the COVID-19, that you develop immunity. What's the truth in that? And then two, you mentioned the examples you're giving about blood clot. You specifically said women. Why women?
Okay, so I'll quickly go through that. It's an interesting question on if you have been infected and you got well, can you get sick again? Before I answer that question, I forgot to make one point really clear. Tom and I are not doctors, are not medical doctors trained to tell you what to do when you have COVID. And if you've noticed, we've stayed away from that. Some people say they are taking herbs and others say they take themselves to hospital and all that. There are WHO guidelines on treatment of COVID. I remember when I was at the Nairobi hospital, some of my friends brought me dawa concussion and they were turned away at the gate and told, no, we're not taking this. So I'm not talking here today about treatment of COVID-19 because I'm not trained in that. However, on the question that you have asked, if you have had COVID-19 and recovered, it is highly likely that you are protected against COVID-19 because what has happened is that you have had a natural infection and you have developed immunity to more than one molecule of the virus. Because remember, when you have a natural infection, you have the whole virus. When we vaccinate you, we only take one piece that we think is the most important to reduce the severity of the infection for you. But when you come into contact with the whole virus, you have a mix of everything, and you have what we call B cells and T cells and all other memory cells have developed, and it's highly unlikely that you are going to get severe COVID-19 again. It's very important to underline that. Now, I need to point out factually also that it is not true to tell patients who have recovered from COVID-19 that they will become susceptible in 3 or 4 months' time. It's not scientifically true because protection for them does not depend on the level of antibodies that they have. It depends on how quickly B cells, sorry to get complex, will produce those antibodies quickly when you have the second infection. So normally you'll get vaccinated or you'll get sick, your antibodies will go high as you clear the little protein. If you have been vaccinated, they will come down again and sit quietly because you don't need them because the molecule or virus is not there. When you have the virus coming up, you have cells that produce the antibodies called B cells, and they will do this, they will multiply very quickly and produce antibodies very quickly so that you'll be able to control that infection. So technically, it is not true that after 4 or 5 months, you can get sick again with COVID-19 because your antibody levels have gone low. That is why we vaccinate kids for life against things like smallpox, things like measles, things like rotavirus, polio, and the immunity is lifelong because it's not that they have high antibodies all the time. Polio, for example, it is because they have a subset of cells that are called memory cells that sit there, and when polio comes, if they ever come into contact with it, they explode very quickly and produce antibodies. So I would say in a nutshell that if you have had COVID-19, it is highly unlikely that you'll have severe COVID-19 again, but most importantly, unless it is a different variant from the one that you had, it might cause some mild disease in you because you have not seen that particular immune system has not interacted with the patterns on the new variant. And so, yes, you could get COVID-19 again if it's a different variant, but it is highly unlikely that you would get severely sick from COVID-19. So we can develop herd immunity through vaccination, or we can develop herd immunity through infection where we all get sick and recover. The problem with through infection is that many of us then die because not all of us have the same level of immunity. That's why we'd rather get herd immunity through vaccination. On the question of women, the truth is that we do not have any idea. This is still under research. The only thing that has been observed for this very little number of people who developed these blood clots after vaccination with the AstraZeneca vaccine is they were women and they were all under 55 years of age. The vaccine, remember, was trialed. There were a lot of trials which were more detailed before licensing. None of these clots were observed in those trials. And this has been rolled out, and then you suddenly have these reactions. I hope this is helpful. Thank you.
Thank you. So I'm seeing Eldamuga put his hand down.
Yeah, just a quick one. Huh?
Yes, please.
Yeah, thank you, Tom. Thank you, Musa. That is extremely useful and informative. I'm almost changing my mind that I need to go to get the vaccine. I just a very quick question. How long does this vaccine keep you safe? I mean, so do you have to get, vaccinated every year, like for instance, in the US, where the normal flu vaccine is a yearly event? And then secondly, in terms of variants and then the mutation, why is it that this COVID is having so many waves, so many mutations? I mean, the common flu really is just a common flu as we know it. But this one, we are now down third wave. What is the possibility we're having a fourth and fifth one with the various, with the virus mutating into, subsequent viruses? Thank you.
Great question. The common flu is not one virus. In fact, you never suffer from the same virus causing the common flu. The common flu is influenza, and it is lots and lots of mutated influenza viruses spreading through the population. It first appeared in 1915 and caused all those deaths, and went through exactly what COVID-19 is doing and settled among us without killing us because we developed herd immunity and started to live with it. So you never suffer from the same variant of the common flu twice. You have lots of variants, and that's why year in, year out, we talk about flu season and we get sick and we sneeze, and some of us might just go to hospital, sleep in bed for a few days, and then go about our business. So the common flu is many, many different forms of the same influenza virus, which have mutated and gone through the process exactly as COVID-19 is doing. What COVID-19 virus is doing is not new. It is exactly how new viruses getting into a human population behave. They change, struggling to try to avoid the immune system, and a point reaches when you have so many variants exploding everywhere each that resembles some of the others that have been there. And then our immune responses are able to respond better. And then generally speaking, we all become covered. The reason why you're hearing this crazy panic around new variant in South Africa, new variants in the UK is because this is the beginning of the pandemic. And this is the most studied pandemic in history. If you were to do exactly what you are doing, study the influenza pandemic in 1915 to 18, exactly the way we are studying it, it would be exactly looking like this. So I just really want to allay people's fears that because of the presence of variants avoiding our immunity, we are going to die, it's over for us, whatever we do with vaccination will not survive. That is actually not true. What is happening is that the virus is racing against our immune system. Our immune systems are racing against the virus. We'll get to a point where we will coexist, which should be in the next 10 years, 8 years, 6 years, we will all be happily coexisting with this. It will almost become like a common flu. On the question of vaccination against the common flu, I don't know what Tom has to say. I will be honest and say that I'm not exactly convinced that vaccination against COVID-19, I mean, against the common flu is useful for healthy people like me and you, especially if you're under 60, under 55, it comes and goes. But if you look at the US, for example, where you have people who live a little bit longer, crowded in homes together, when the flu comes and they have all these underlying conditions, it helps for them to have a vaccine that most of the time gives them a little form of immunity, but I wouldn't necessarily say that their lives really are dependent on that vaccine. Many people don't take the common flu vaccine and survive. And the reason why we vaccinate every year against the common flu is because of the variants, exactly like we are saying. But what you get as a common flu vaccine in the US today is the flu that circulated in parts of the US 2-3 years ago. So it was isolated and processed, and the companies prepared it, and 3 years later, they're giving it to people. It's likely that those variants are no longer in circulation and you have new variants. So technically speaking, the usefulness of the flu vaccine, personally for me as a scientist, sits between business and benefits, especially for specific age groups of people. I never get vaccinated against the flu personally. I'm not saying you shouldn't, but I think that if you're vulnerable, immunocompromised, if you're aging, it's important to take it. But the truth is that all these variants are there. They start looking like each other with time, and they get to a point where they coexist with us. Thank you.
Thanks, Musa. I'll not add to that, since you've made the comment around pharmaceutical companies updating the flu vaccines on an annual basis.
Yeah.
So why people take every year is simply to keep the immune system as current as possible. Yeah. So just that. I see more hands up. Patrick.
Thank you very much. I've enjoyed the talk, and I'm not an Adventist. I'm here courtesy of Engineer Kidenda and Mary Claire invite. I'm a Catholic Baptist, but I really liked the opening about religion, faith, and science. Thank you very much. Let me start from where the lady left. These clots in women, have they only been observed in Europe or other parts where this vaccine is being administered, like in India? And has there been any in Africa? Maybe not. For my second question, is this vaccine safe for persons with pre-existing conditions such as asthma or any other commodities? Maybe you can just shed some light on that. Number three, this South African variant, probably it is here in Kenya. I had on some discussion today morning on BBC that the AstraZeneca vaccine is probably only 10%. The efficacy on the South African variant is probably only 10%. If that is correct, then how does, I know you've explained it, but maybe you can just, how does the vaccination of using the AstraZeneca vaccine really protect you against the South African variant. Number 4, handling of the vaccine is under refrigerated conditions. What happens when you administer vaccine which has been out of the said conditions of registration for refrigeration for a while? Does it still maintains its efficacy, or does it get compromised? Then the last one, what happens when you get the COVID disease between the first vaccination and the second one? Meaning you get the first vaccination, then before you get the second one, you catch the disease. How do you explain those circumstances, and what happens to your immunity and the protection of the vaccine in those circumstances? Thank you.
Thank you, Tom. I'll let you take the question of the AstraZeneca vaccine and its protection that is being, Patrick says he had is 10%. I'll also let you take the refrigeration part of it. And then I will take care of the pre-existing conditions, the women in Europe, and COVID vaccination in between the first dose and the second dose. So Tom, go for it.
Okay, I hope I'll not stray into some your question. So let me start with the question around the South African variants. Yeah, that's one of mine, Musa. Yeah.
Yes.
Okay. Yeah. So it is true that there is a mutant virus in South Africa. I think there's another one in Brazil, and another which is in UK. Now, South Africa government stopped the use of AstraZeneca vaccine after finding that it's not quite effective against their local strain. Now, there are some early stage studies which have happened. I read one recently where they showed that in vitro just means outside a living system, yeah? They isolated antibodies following vaccination by the AstraZeneca vaccine. And in a laboratory, they checked whether these antibodies bind on the South Africa strain. And they showed that indeed they bind, but subsequent studies need to be undertaken to see whether in living systems this is the same. So what I'm trying to say in a nutshell is that the knowledge is still quite limited as far as the cross reactivity between the AstraZeneca vaccine and the South African strain is concerned. I think for now we are safer to take the South Africa Ministry of Health word at it that the AstraZeneca vaccine is not very effective against the strain that is circulating in South Africa. Yeah. So the next one I was tasked to handle is
refrigeration.
The refrigeration ones. So in theory, vaccines they lose their efficacy very quickly. When they are outside the refrigeration parameters that they've been designed to be kept under, and it varies from vaccine to vaccine on how long it can stay outside refrigeration. Yeah, I am not sure, Musa, what the parameters are for the AstraZeneca vaccine. Is that published information? I'm not sure, to be honest.
So I have shared the parameters for the South African, for the AstraZeneca vaccine, which I think is 4 degrees.
Yes.
In the regular fridge, Moderna is -20, Pfizer it is -70. And for Moderna, they specify up to 6 months. The point, I think, is when Tom says they lose their efficacy, we are talking about molecular structures that are intact. And we are talking about for RNA, it's a molecule that really will not sit at room temperature here and still stay intact. And when we talk about vaccines and an immune response, we are talking about response to specific protein sequences, or if it's RNA, it is RNA that will be translated into proteins. How it will be translated in your body depends on how stable it was. If it degrades and you have that structure broken up, then of course you do not have a response. So just in a nutshell, the reason why it is highly unlikely that refrigeration, if you remove the vaccine from refrigeration for an extended period of time, and we need to look at studies saying if you remove it, it has to stay out for 2 minutes, 5 minutes, whatever, and then it loses efficacy. The ideal is get it out of the refrigerator, put it in the needle, and get it into somebody as soon as possible. That's how it should be. I'm not going into the details of how quickly that is happening. But typically we should have people trying to look at stability studies for those and getting comfortable that what is getting into people is still intact. On the question of pre-existing conditions, I'm not a doctor, but for most of this, I was looking at the CDC site earlier today, and they are very specific that if you have pre-existing conditions, if you're immunocompromised, if you are this, tell your doctor. And I think that there are conditions around if you can be vaccinated with any of those vaccines or not, based on studies that have been done so far. Generally speaking, there seems to be a good picture around pre-existing conditions for most people, but you'll all appreciate that this is new. Data is still coming in for pregnant mothers. We are not sure for certain conditions. I guess information has to come out with time as we try to control the pandemic. And then
Sorry,
yes, please.
On just that point, I think just this week, data was published on phase 3 trials of the AstraZeneca vaccine from US, which showed that the vaccine prevented the clinical disease even for people who are over 65 and people who had other comorbidities. Mm-hmm. But Musa is right, it's a new disease, it's a new vaccine, and more data is still coming, but early indications are very encouraging. Thank you.
Yeah, and then finally on the question of COVID-19, getting COVID-19 in between vaccination, it's an interesting, good question, because in some countries, what's going on is, in fact, people are being given one dose first as an approach, and then they were going to receive their second vaccine after 3 months. I think my wife got hers last month. She will get the booster in June. And there have been a few studies showing that even with the single dose, you still are able to, I think it's about 60% of people seem to be protected just from the first dose. So I think that it is great to even have just the first dose of the vaccine as you wait for the second dose. If you get COVID-19, it is likely that you will still have some measure of protection. Thank you. Tom, how many more questions are we fielding, and how many people?
I think let's take, I'm seeing Galaxy S10, Dr. What's the name there? Sorry. Someone has raised their hand. We take that one. Yeah. Then we take a call after that.
Okay.
I know also the time is really. Yeah. Please go ahead.
The Galaxy 10 this is Dr. Onyanja. Thank you so much. I found you in the middle.
Thank you.
I found you in the middle, so I did not get how you started, but I'm told you were able to bring out the region science and the other part, so I didn't get that one. But it has been very exciting. I got where you were explaining how the virus, the RNA and DNA work. Wonderful, and I hope and I thank you, because you have really simplified for many people. Hopefully they were able to understand. I hope I was not enjoying because I am a doctor. I hope that they were able to catch the rest of the people. But I just have two questions. One, you said you have studied the people, the number that received vaccines the 5 million and then the 11 million, where we had 30 and 11 people, women. But I don't know whether you have looked at, for example, in the general population, we know there are people who develop those blood clots with or without the vaccine. I think what might bring out something that will help us is that if you took 5 million people in the general population without vaccination, how many will be likely to have the blood clots? Because I know people develop blood clots. That may also help us to see, is it a coincidence that among us, like for example, the 11 women in the 11 million was something that somebody could have developed, not necessarily because of the vaccine. I think that looking at the prevalence of clots in the general population will be able to help us. Secondly, I'm just wondering that can we say literally that a wave is because of a new strain, given what Elder Muga was talking about? Is like we are the virus has mutated, that we are in the third wave. Are the waves equivalent, or are they the same as saying that we are having a new wave? Or I'm looking at a wave, for example, in Kenya, if I take the third wave is actually finding us because most people laxed and there was a lot of transmission without actually being careful. And now it is now exploding after having maybe slowed down in terms of our the IPC practices, the social distancing. I want you to comment the relationship between the variances, the strains, and the waves. Thank you.
Thank you. Very quickly on the first question, I actually tried to look at the incidence expected of blood clots. I have to go back and confirm, but the comments, information seemed to be saying in normal population, you expect 5 people. I think there's a proportion there we'll look at, but there is an admission that there's a ratio of 5 people, and I'll get the denominator number that we're measuring this against. But it is 5 women that are expected to have this. But for the population that has been vaccinated, it seems the incidence is 12 women, proportionally around ratios. And I will look at that. So there is admission that for some reason, there is an increase in the number of women getting these blood clots out of the normal baseline. If I've understood your question, I agree with you that that is important. And so they are still trying to find out why is this the case. And I think the previous caller also asked, is this just in Europe? So far, the data seems to be in Europe only. I don't think India has reported this or Africa. But at the same time, how well do we, monitor these things? On the question of waves and new strain, my personal suspicion is that what we are reaping now is the result of the Christmas holiday, where we went home and everybody, travel was open, and then people came back, and then what we are having is a slow multiplier effect of people who got infected at that time, infecting others, and then you're now having more and more people getting COVID-19. I think that is probably the reason. We are not doing in Kenya any serious surveillance of the variants. I work at ILRI, we have a testing center at ILRI, we test people. We are only keeping the viruses that we are isolating and hoping to do some sequencing to see whether this is a new variant or not, which will be the result of years to come that we can publish. But I'm not sure that we have a government of Kenya system now. And you can tell me, Dr. if we do, that really is on top of variances. When I was at the hospital, we were quite a number of us, and there was quite an international mix of people there from the UN system, and I did not see anybody. We are only keeping the virus.
Yeah, if I may interject, that
yes,
yes, we are not doing it at a big scale, but I think a small scale kind of sequence was done, which really, I think, I can't remember exactly, but I know we had some of the strains from South Africa. The majority was actually the one which we had from the beginning. So something is being done, but not in the big scale.
But just to answer you, I don't. We have no way of confirming that the wave is because of a new variant. I think it's
Yeah,
not easy
that's
to
a good
tell,
point to
and
make, yeah.
it's not possible to make that correlation without having the data to do that.
Thank you. Allow me to just
I
make a
think,
comment.
Musa, just to, sorry,
can I
there's
make?
a,
Yeah.
one second, Daktari. There's a bit of noise on the call. Can we be on mute unless we are talking? Daktari, go ahead.
Yeah. So what I why I am I'm just concerned about the issue of the strains and the wave. I think there's a bit of misconception that we are having actually a new strain, and that is why maybe it is so bad, and people are now worried that even if I go for vaccination, suppose another strain comes. I agree with you that we are actually reaping the results of the Christmas and the many gatherings, political and all that. And I think the moment we understand that that is the reason, then actually the people, Kenyans, will be able to say, we are actually responsible for our own actions, so that we are able to follow the protocols as given, not hiding behind that, no matter what I do, if a new strain comes, it will come with another wave. I think, to me, if that can be very clear, then it will help us. Even people will have a reason to go for the vaccines and a reason to be able to adhere to the protocols. Thank you. Over to you.
Yeah, thanks. And I think it's fair to assume, I think these waves also tie to the point Musa made on herd immunity. So by herd here, it means HERD, like a population of cattle. Yeah. So the big take from herd immunity is that once a big enough population gets exposed to the disease and they build immunity, then the disease has nowhere to go. So you find the small population which remains unexposed gets protected, just because in the population a majority of people have been pre-exposed. Now the hard way is for the disease to just run wild and infect most of the population. Yeah. The easier way is to vaccinate. Yeah. So if we vaccinate a good chunk of our population, then the disease has nowhere to go. Then you come to a point where even the population which has not been vaccinated or has not been exposed to the disease, naive individuals, they are protected from new infection. So these waves are simply, the disease is spreading to new individuals who are not immune before. And my fear is that we will have a fourth wave also, until a point where this thing stabilizes. That's my fear. Epidemiologists are putting the herd immunity for COVID at about 67%, but we all know that this point, which we call herd immunity, also has inertia. So it might go all the way to 80 before we realize the benefits of herd immunity. So the point I'm making is that let's try to protect ourselves as much as possible. If you can access the vaccine, yeah, please get it. I know so far we have 1 million doses which have been imported. So the most vulnerable groups are being given, or the high-risk groups are being given this. But for the rest of us, I think it's up to us to take individual care. It's we don't all want to contribute towards this herd immunity. Yeah? Thanks, Musa. You're on mute.
Yeah, I don't know if we can finish or if there's anything else. I'm happy to discuss this and get in touch. There is so much going on social media about this topic, and people are speaking from a position of no knowledge. It would really be good even if the messaging around this is taken up by the ministry and framed in a way that people can understand and follow and make reasonable choices about what they want to do. But the default for most people is they listen to these inflammatory messages from social media and quickly, oh, this is the new world order. They're putting the mark on the beast on me. I'm not going to be part of that. So I think that there are probably other people talking about some political and other dimensions of birth control and things like that, that I'm not sure they are rooted in fact, but I'm only saying that it is important that we get the right information and make the right choices.
Thank you. Thank you. So I think we are done. One sec. Sorry. I'm with Elder Mabea, and he has just received, I think, a text. Someone is asking whether if they have been infected with the disease, they need to be infected. Musa, you handled this question earlier.
They need to be vaccinated.
Yes. You did handle this question. If you can just run through it very quickly.
Yeah, very quickly. If you have been infected, let's say there is one dose of vaccine left in your family that you have access to as a family, and the husband has had COVID and recovered, and the wife has not had COVID, I'd rather the vaccine goes to your wife than to you, because you have a measure of immunity that I can reasonably say will last for quite a while to the same variant that you have been vaccinated with. And also be careful to recover from COVID and then go for vaccination a month later. I think there is a period that you need to take. You don't want to overexcite your immune system with a vaccine of something you are recovering from. So you need advice around when you can take it. But I don't think that you should kill yourself to run and get vaccinated if you have recovered from COVID-19.
Yeah, that's correct. You have some immunity for the same virus which will last. And this talk around antibodies disappear after 5 months, therefore at that point you'll be vulnerable. It really, as Musa said earlier, in immunology terms, it's what is really important is the memory. Yeah. So the antibodies can go down. Yes, I think recent studies have gone as far as 8 months and they are still finding circulating antibodies. But the point is, at the point when they subside, you are what we call B cells. They have a memory, and when they detect that that protein is in the system, they respond very quickly to produce antibodies against that. So I think we will leave that at that. The good thing is that we have also recorded this presentation, so it can be shared and people can come back at their points of convenience and just watch what was being presented and discussed. So at this point, I'll really want to thank Musa. I think, Musa, you've really helped us with this presentation. I know it must have taken you quite a while to put it together. Lots of reading just to generate these 3 slides. So we really appreciate you taking time out of your busy schedule to prepare for this meeting. May God really bless you.
Thank you. And Tom, did we pray when we started?
When we started, we prayed. Yeah.
Okay.
When we started, we prayed. So in finishing, I just see Pastor, our church pastor, Pastor Oyamo, is on the call. Pastor, I'd like to hand the program back to you to give some closing remarks and close for us with a word of prayer. Thank you. Pastor, are you on mute?
He seems to be on mute.
Let's wait for him for a second. Oh, he's here with me. I just realized he was on another device. So I'll just put on the side here and allow Pastor to close.
All right. Thank you so much. Thank you so much, Dr. for the work that has been done. And I must assure the congregation watching, those on YouTube and on the other social media platforms, that this message is here to stay. We want everyone to watch it. We want to share it all over, that we may get to know this. Personally, I have been blessed by my at least glimpse into the scientific world of how this virus is, and all the things that have been shared with us through Brother Musa. Brother Musa, thank you so much for coming, and we welcome you to join us again some other day. If we develop more questions, we would still call on you. And thank you, Dr. Sebe, for also organizing this. Allow me to pray so that we can come to an end. Dear Father and our God, we come to you now. We want to thank you so much because of this day. We want to thank you for the privilege that you have given us through your servants to get a glimpse of this virus and also of the vaccines that are out there. I thank thee for your sons and daughters who have participated in this, and those who have watched and those who will be watching also endlessly according to time, that they may be blessed of the same. You who speaks to your people, may you also speak to us to strengthen us and encourage us during this pandemic, that Father, we may come out of it stronger than this day. And I ask in a special way that your spirit that speaks to your children all over the world may keep us in comfort and to help us prepare ourselves for your soon return. And until then, oh Lord, guide us in the ways of righteousness and help us work according to that which you have called us unto. And may God bless us all and be with us, all who have been listening. For these things we ask in Jesus' name. Amen. Amen.
Thank you.
Thank you so much.
Okay, thank you, everybody. I think we'll close the meeting here. All right. Bye-bye, and have a blessed rest of the day.
Thank you. Bye.
Bye. Thank you.